So asexual cell division, this is where a cell is going to make identical copies of other cells. So this is occurring in your body when your cells are repairing themselves and when they're growing, when you're going through growth spurts, when you're regenerating new skin cells, all sorts of things. Interphase is the first part of this asexual cell division, and we've learned that interphase is G1, S, and G2. In G1, we're just growing. The cell is just growing, getting bigger. In S, you actually have a doubling of the DNA because the cell is saying, okay, I'm getting kind of big. I need to manage my space here. I need to double my DNA to get prepared to divide in case I get too big. And then G2 is your second growth phase where it's at the kind of the end of its life cycle and ready to divide into two new cells. When you look at what the DNA starts as, when we're looking at a human cell, it's 46 chromosomes. When we're looking at this cell here, because we don't want to draw 46 in there, we have 4 single-stranded chromosomes. So at the beginning of G1, you're at 4 single-stranded chromosomes. Then what happens is the DNA will replicate in the S phase. And in the S phase here, these 4 chromosomes will each make copies of themselves. And those copies that they make of themselves will join together with the original, bind together by the centromere of the chromosome, and now become 4 double-stranded chromosomes. So if we watch the animation, and hopefully this guy won't continue to talk here like he was when I first tried this. If we just watch, the guy is talking and you don't know yet, but it's showing you what's happening inside the cell at the time, showing you that there's the nucleus, there's the centrosomes, and what will happen right away here is the DNA is going to replicate. So if you just follow this little box on the left-hand side, and you'll see those 4 single-stranded chromosomes, we should see those doubling into 2 chromosomes right away. And there we go. So you've got this, and I'll pause it. We've got now the S phase occurring. So the cell has just been living its life cycle, realizing it's getting kind of big, maybe getting close to having to make 2 new cells, 2 identical copies. And so the only way you're going to get identical copies of cells is having identical DNA in the cells. And so each DNA strand makes an identical copy of itself, and those copies pair up with their original to now make 4 double-stranded chromosomes. So when we're counting through the chromosomes, I went through 4 single-stranded chromosomes, and now I'm at 4 double-stranded chromosomes. Those double-stranded chromosomes are composed of what we call sister chromatids. So if you look down here at the bottom, I'll circle it in green, right? You see those chromatids, those are sister chromatids to each other. So when the original pairs up with its copy, we can call that a sister chromatid. So if we watch what happens as this process is continuing on, G2 will happen. And in G2, the cell is just going to continue to grow again. And those chromosomes are going to condense and actually look more like chromosomes. Because really in interphase, they don't look like this. They don't look like what they have drawn on there. They actually look, if you look inside the cell there, they look like a bunch of spaghetti noodles. So those spaghetti noodles during just the start of interphase, or just the end of interphase, sorry, are going to start to condense into chromosomes. So now we're entering a prophase. Okay, so I'm just going to scroll back a little bit because this guy went a little bit ahead. So here's prophase. So during prophase, and we're getting interrupted by the intercom. So during prophase, you have this spaghetti-like chromosomes that are now condensing into what we see as chromosomes, these X-shaped structures. You've got your centrioles moving towards each end of the cell, and then what will happen to those condensed chromosomes is that they will all line up at what we call the equator of the cell, or halfway down the cell. When they line up at the equator of the cell, we now call this metaphase. You'll see that they're calling it prometaphase because it's just before metaphase, before they're actually lined up in an individual row. Once they're in that individual row, we can officially call it metaphase, and there we see them all lined up nicely, just like you did back in elementary school, lining up to get back in, right along that equatorial plate. So we continue along during metaphase, and then what happens is those sister chromatids that were pairs, identical copies of one another, are now going to get to be pulled to opposite sides of the cell. So now we are turning into single-stranded chromosomes again. So we started as 4 single-stranded chromosomes, we doubled to make a copy, and then now the cell is going to split into two cells that have 4 single-stranded chromosomes. Well, that's what we started off with in the first place. Okay, so, and those were all copies of the original, so if we scroll all the way back, we're back to where we started, and we've made a copy of that original cell that we started with.